The next part of our presentation today is going to be a series of talks by researchers who are out on the frontier trying to discover what is EHE, how does it work, what is the underlying biology that will reveal to us ultimately what treatments might work and allow us to test those treatments to figure out if they're going to work. This research is helping move things forward and is a prerequisite to getting us to a place where this disease can be treated. We're going to start off the session today with Dr. Lamar.
Great. Okay, so I hope everybody can see the screen and hear me. I just want to thank the organizers and the EHE Foundation for inviting me to participate in this amazing meeting. And so what I'm going to tell you about today is a little bit of the work from my lab aimed at trying to inhibit the tumorigenic function of this TAS-CAMTA1 fusion protein that you've heard about. And we work mostly with this TAS-CAMTA1 fusion protein, but a lot of what we've discovered so far we think will also be applied to the YAP-TFE3 fusion protein as well. And I'll try to show you that throughout the course of the talk. So as we all know, the vast majority of all EHEs contain either the WWTR1 CAMTA fusion gene or the YAP-TFE3 fusion gene. And so these fusion genes are a result of something called a reciprocal translocation. And so this happens, and just tell you a little bit more about how this happens. So basically, you know that genes are contained in these linear pieces of DNA in the cells, and they're organized into chromosomes. And when a translocation happens, what happens is part of one of these chromosomes can be exchanged with part of another chromosome. And so the resulting chromosomes have essentially exchanged arms. And this is what happens in EHE. And so just to give you sort of an example of what this looks like, these are cartoons showing the TAS protein, which is encoded by the WWTRE gene, and the CAMTA protein. And so when this translocation event happens, the beginning part of the TAS, the gene that encodes the TAS protein, becomes fused to the end part of the gene that encodes the CAMTA protein. And so you get this new gene that encodes a new protein that has parts of both of those genes. And it has now sort of new functions because it's also missing parts of those original proteins. And so we now know from research from a number of groups that these two oncogenes, or these two fusion proteins, are oncogenic, which means that they can cause cancer to occur. And so this is just some data from my lab to help you see that. And so what the graduate student in my lab, Ryan Kanai, did was take the gene that encodes the TAS-CAMTA1 fusion protein, and he expressed it in mouse fibroblast cells. And then he injected those mouse fibroblast cells into mice, and he also injected control cells into mice. And basically only the cells that had the TAS-CAMTA1 fusion gene being expressed in them could make tumors in the mice, and virtually none of the mice injected with controls formed tumors. And this is consistent with a lot of the published publications that have come out where other groups have shown in this fibroblast model that TAS-CAMTA1 and YAP-TFE3 can drive tumor formation, and also with the new recent studies in the genetically engineered mouse model and transgenic mouse models from Brian Rubin's lab and DJ Pan's lab. And so we know that these are the cancer driving events in EHE. And so just to give you kind of a very brief overview of what we know and the role that these fusion genes play, I'll remind you again that the translocation happens in a cell in the body. And so then these fusion genes are generated, and then that cell will begin to make the protein that is encoded by that fusion gene. And we know from previous studies that these fusion proteins do not respond to the same regulatory pathways in the cell that would normally repress the activity of the TAS and YAP genes. And so they're essentially inappropriately active versions of these proteins. And so they travel to the nucleus of the cell, and they partner with another family of proteins called the TEADs. And this complex now will turn on or off the expression of other genes in the cell. And by turning on and off the expression of genes, I basically mean by increasing or decreasing the proteins that the cell makes from those genes. And we think it's these changes in protein expression in the cell that are causing the formation of EHE and allowing the EHE to grow into a bigger tumor. And so with this in mind, how do we approach trying to treat EHE? And there are a couple ways we can do this. My lab is really focused on trying to block or inhibit the ability of these fusion proteins to turn on or off these other genes. And there's a couple ways that we think about trying to do this. The first is to prevent the cell or to reduce the levels of the fusion proteins that the cells have. And so you can do this either by preventing the cell from making the fusion protein or by causing the cell to degrade it after it's been made. Another thing that we can do is try to block the cell, the ability of these fusion proteins to travel to the nucleus. And then also we can try to block the ability of these fusion proteins to interact with this other family of proteins called the TEADs. Okay, and so my lab is really focused on trying to get these three processes to happen in the cells. But of course, you can also think about treating EHE in other ways as well. And that involves trying to identify some of these other proteins that are being regulated by the fusion proteins and block their function, as well as just treating cells or patients with drugs that will kill the EHE cells or at least prevent them from dividing. And that may occur through a pathway that's completely independent of this pathway, but still be an effective way to treat the disease. But again, we're focused really on trying to block the function of these fusion proteins. So that's basically the goal of this initial work in my lab. And as part of that goal, we are also trying to develop EHE model systems that we can use to test some of these pathways that we identify. And I'll tell you about that very briefly at the end. So the strategy that we're using to try to identify regulators of these fusion proteins uses a technique that researchers have used in the past. And it essentially borrows some biology from the firefly. And so what we've done is we've taken the enzyme or the gene that encodes the firefly luciferase enzyme, which is the protein in the firefly that allows them to emit light. And we're going to put this into cells in such a way that it only gets produced by the cells if the cells have a lot of TAS-CAMTA1 TEAD activity. And so if the activity of this fusion protein is very high, then you'll get lots of this firefly luciferase enzyme being made by the cell. And so then we can just give the cells the substrate for this enzyme, and they will emit light. And the amount of light that they emit is going to be directly proportional to the amount of TAS-CAMTA1 activity in the cells. And so the way that we actually did this experiment was we took the mouse fibroblast cells again, and we stably expressed the TAS-CAMTA1 oncogene in them that we got from Brian Rubin's lab. And then we took those cells and we put them over a bunch of wells in a dish, and we put in this firefly reporter assay that I just described. And then we take and we alter the expression of proteins that we think might be repressing or activating TAS-CAMTA1 in each of these wells. We allow those manipulations to take effect, and then we give the substrate for the firefly luciferase enzyme to the cells and measure the amount of light that the cells are emitting. And again, the cells that have high TAS-CAMTA1 activity, which would be acting like tumorigenic cells, will emit a lot of light. And then any of the manipulations that we've done with the various things that we've added, if they reduce the amount of light emitted by the cells, then that tells us that those manipulations have repressed the TAS-CAMTA1 fusion protein. And that's what we're looking for. And so Ryan Kinases, the graduate student in my lab who's been working on this, and when he did this experiment with a bunch of candidates that we wanted to test, he identified 7 proteins that could repress the activity of this TAS-CAMTA1 fusion protein in cells. And I don't have time to explain what all these proteins do, but I will tell you that these 4 here were already known to regulate the normal TAS and YAP proteins in other contexts in cells. And they do so by affecting part of the protein that is contained in the fusion gene. And so we think that they're probably affecting TAS-CAMTA1 activity in the same way that they affect YAP and TAS activity. But we still haven't tested that, which is why we have question marks there. Ryan also identified another gene called calmodulin, which we think is actually binding to the CAMTA portion of TAS-CAMTA1 and preventing it from going to the nucleus. And so we've just obtained some additional funding from the USA and Australia EHE foundations to pursue this gene more rigorously. So we're kind of excited about that. And then I'll just also mention 2 other ones that we identified called the AMP kinases. And so these are also known to regulate the normal YAP and TAS protein, and they do so by preventing it from interacting with the TEAD family of proteins, which is, as I told you, one of the strategies that we're looking at. And so we think that's probably how it's regulating TAS-CAMTA1. But I will also mention that these proteins are known to inhibit another protein that might be important in EHE, and that's the mTOR protein. And this mTOR protein is able to activate YAP-NTAS under normal circumstances. And so by inhibiting its function, you're effectively inhibiting the YAP-NTAS proteins. And so we think that that may also play a role in its regulation of TASCAMP1. And I will also mention that the reason we became interested in these proteins initially is because there are FDA-approved drugs that can activate the AMP kinases, which we think would, again, repress the TASCAMP1 fusion protein. And there are also available drugs that can block the mTOR protein pathway, which, again, we predict would repress this TASCAMP1 fusion protein. And of course, you just heard that some of these mTOR inhibitors are used already to treat EHE patients, and we've seen some nice success with some of them in some patients. The metformin has not been formally tested in EHE, but there is some anecdotal evidence from patients who go on metformin to control their diabetes and have found that it can actually stabilize disease. And so we don't know if those clinical results have anything to do with this very preliminary data that we've found, but it's interesting to think that maybe some of these drugs are actually directly inhibiting these fusion proteins. And so moving forward, that's what we're trying to do. We're trying to understand how these different candidates that we've identified are regulating the fusion proteins. We do plan to test some of these drugs in these model systems. We also predict that many of these same genes will repress the YAP-TFE3 fusion protein, so we're going to test that next. And then we want to develop some EHE model systems to actually confirm that some of these same candidates can regulate these fusions in EHE cells and block their tumorigenicity. And so I'll just spend the last couple of minutes real quickly telling you about that. And so through our collaboration with the Rubin Lab, we've obtained the mouse EHE cells that Caleb Seavey generated from their genetically engineered mouse. And so Ryan's been in the process now of kind of optimizing some of our common assays that we use to study TASCAMP1 in the fibroblasts, as well as to study the normal YAP-NTAS proteins in other cancers. He's optimizing them so that we can use these EHE cells and start testing some of those candidates with these cells. We're also trying to generate tumors that we can use to test some of these candidates and the effects they have on an EHE tumor growing in a mouse. And for this, we're using a technique that was developed by a graduate student in my lab for studying other cancers. And so just quickly, what we do here is we take cancer cells and we label them with a fluorescent protein, which allows us to see the tumors and the tumor cells in the mouse. We also put in that same firefly luciferase enzyme, but this time it's being expressed constitutively or all the time by the cells. And I'll tell you why in a second. And so then we use an ultrasound to guide a needle injection of these cells into the mouse's heart. They spread throughout the entire body. And when we do this with other cancer cells like melanoma cells or breast cancer cells, we see lots of tumors that form in the liver, lungs, bone, and other tissues of these mice. And of course, liver, lungs, and bone are three of the main sites that EHE form in. And so we think we can use this technique to generate liver tumors in these organs and study them. Because the cells have this luciferase enzyme in them, we are able to track the growth of these tumors in a living mouse over time in a non-invasive way. And so what we can do is allow these tumors to form and then target the candidate pathways that we've identified or treat the mice with drugs and inhibitors and ask what happens to these tumors over time. And hopefully we will see that they either stabilize or start to regress, get smaller. And then we can also ask, is the response that we see the same if the EHE is growing in a liver or a lung or a bone? And that can allow us to start to distinguish how well some of these treatments may work on different types of EHE. And so we've only just begun this study now. We've injected the first cohort of mice. And so just to see how things were going, we took 2 mice at a very early time point just to see what we could, if the tumors were forming. And indeed, we did see that the EHE cells were forming tumors in both the lungs and the livers of both those mice. And they're still quite small, but with time they should grow. And so we think we have a nice model system to start studying some of our candidates in mice. And so hopefully we'll have more to present on that in future meetings. And with that, I will stop and just thank the members of my lab who've done this work. And it's mostly driven by Ryan Kanai, who's the graduate student who's working on EHE. Got to thank the funding. And so our initial work on EHE was made possible by funding from all 3 of the EHE foundations in the UK, US, and Australia. And then, as I said, more recently, the US and Australia foundations are funding that project on that one gene that binds to CAMP1 that I mentioned. We also have funding from the EHE Collaborative Program at the Cleveland Clinic, which is funded by the Margie and Robert E. Peterson Foundation. And none of this work would be possible without the generous support of Brian Rubin and the members of his lab as part of our collaboration. It's been a real amazing collaboration, and they've been extremely helpful to us, so we're very grateful. And then finally, I just want to thank Guy Weinberg, because it's really Guy who got me interested in EHE in the first place by inviting me to participate and co-organize the Telluride conference with him. And so I'm grateful for him and his foundations for their support. And thank you all for your time, and I look forward to answering questions later.
Thank you much, Dr. Lamar. Our next speaker will be Dr. Tanas.
My name is Munir Tanas. I'm a associate professor of pathology at University of Iowa. What I'm going to discuss with you today is just an overview of epithelioid hemangioendothelioma. There's been a lot of discussion today and yesterday about various models and where we're going from a treatment standpoint. And so I'm just going to try to condense that a little bit and integrate some of the findings from our lab with some of the other outstanding work that's been shared today and yesterday. So as we've heard before, epithelioid hemangioendothelioma is a vascular sarcoma. It can occur over a wide age range. There is a predilection for females in the disease. It can arise anywhere, essentially anywhere in the body, soft tissue, bone, as well as visceral organs such as the lung and the liver, where it characteristically presents as multiple nodules. We now know that this likely represents early metastasis. The nomenclature is kind of confusing, but epithelioid is basically referring to the fact that the tumor cells are rounded when you look at them under the microscope. Hemangioendothelioma is a historic term used to indicate vascular tumors of intermediate malignancy. Now we acknowledge EHE as a full-on sarcoma in the latest WHO classification. The neoplastic cells in EHE are characteristically arranged in these cord-like structures and embedded in this myxoid hyaline matrix. What's really interesting about EHE is that even though it's a vascular tumor, when you look at it under the microscope, it doesn't form these well-formed blood vessels. And so we have to rely on other ancillary techniques, or such as immunohistochemistry, look at expression of proteins such as CD31 to confirm its vascular differentiation. And you've probably seen this slide before. It's a really nice reminder that EHE, this is a karyotype of EHE, so these are all the chromosomes that have been isolated from an EHE tumor. And what's nice about this karyotype is it shows, as John mentioned in his previous talk, this reciprocal translocation between the long arm of chromosome 3 and a short arm of chromosome 1, and vice versa. And the genes involved in the pathogenesis, or in the initiation of EHE, are encoded by this chromosomal translocation, in particular the fusion event on chromosome 3. And as has been pointed out before, when you look at the karyotype, the remaining karyotype of EHE, it's really just these two chromosomes that are altered, and the rest of the chromosomes are relatively normal. And so this underscores this idea that it's a relatively simple cancer from a karyotypic or genetic standpoint, and it also lends support to this hypothesis that the genes involved in the breakpoint of these fusions are important to the pathogenesis of EHE. And as Brian shared yesterday, when we identified the WWTR1 Campta1 fusion encoded by this chromosomal translocation, we were fortunate to be able to leverage some advances in technology, in particular RNA-seq, as well as collaborators at Yale who developed this fusion-seq algorithm to basically skip over some of these steps that required the use of an EHE cell line, which we didn't have available at the time, and identify these gene fusions. As you can see, that there are 69 million paradigm reads that were generated by the RNA-seq experiment. Using this fusion-seq algorithm, we were able to pare it down to 200 fusion transcripts. But because we knew the karyotype of the tumor that I showed in the last slide, we were able to sort through these 217 fusion transcripts and identify the WW201-CAMP2 fusion transcript as the genes not only involved, not only present on chromosomes 1 and 3, but the exact bands involved in the reciprocal translocation and fusion. And so, which is basically identifying these genes. It was basically a needle in a haystack type of problem that was resolved by this approach. And so the WW201-CAMP2 fusion was confirmed by RT-PCR. This is the RNA breakpoints on the index case, as well as 4 additional EHEs that we did RT-PCR on. And they show these fusion events between exons 2 and exons 3 of WTR1 and breakpoints within exon 9 of CAMP2. As you look at the breakpoints within exon 9 of CAMP2, you can see that there's some microheterogeneity here. They're not all in the same part of CAMP2, but importantly, the breakpoints here result in CAMP2 being fused in a frame in a particular way to WTR1. So there's a functional protein on both sides of the fusion protein, both the N-terminus and the C-terminus. And so similar work has been done with YAP-TFE3 fusion protein, which constitutes the remaining 10 to 15% of EHE that don't harbor the TAS-CAMP2 fusion protein. The fusion proteins are highly homologous and basically contain the N-terminus of TAS or YAP fused in frame to another transcription factor, either CAMP2 or TFE3. And what these fusion proteins have in common is that they conserve the T binding domain of TAS and YAP, as well as the transactivating domain of their fusion partner and other protein-protein interaction domains. And several labs have elucidated this, including Frank Szyszalski's work yesterday, and the graduate student in my lab, Keith, also presented some work elucidating the function of C-terminus. We'll get to that a little bit later in the talk. One of the obstacles, one of the challenges when first studying EHE, this is about 13 years ago now, is that we didn't really have great models of EHE to dissect the function of these fusion proteins. And so this represents about a year and a half of work I did in the lab, trying to express the TAS-CAMP2 fusion protein in these primary cells or immortalized fibroblasts and endothelial cells, and really was unable to identify a hallmark of cancer shown here with regards to proliferation, migration, invasion, or antigen-dependent growth, except with the NIH3T3 cells. And we published a lot of these findings in 2016. And but NIH3T3 cells have some limitations, and so other models were just urgently needed. And what I really wanted to point out in the remaining part of the talk is just how much progress has been made. It's been really exciting to be a part of this field. There's this fantastic paper that was published in Genes and Development out of Brian's lab. Caleb was the first author who we heard from yesterday. And as you can see over here, that this mouse model used a conditional approach where the WW201-CAMP2 fusion gene was expressed under the control of the WW201 promoter. And interestingly, just to underscore how close these tumors resemble the human tumors, both pathological and genetic approaches show that these mouse tumors were virtually indistinguishable from the human EHE tumors. And this just really underscores how reliable this mouse model is. There's also some fantastic work done out of DJ Pan's lab. Jordan Driscoll is the first author, and they use a tetracycline based inducible expression system for WW2-CAMP2, And in this mouse model, the mice developed vascular tumors involving the lungs, which, as we mentioned earlier in the talk, is a place where EHE can occur, and similarly exhibit this multifocal presentation. As has been shown from multiple investigators in this mouse model, it was determined that delta-campta-1 drives a T-based transcriptional program and was required for tumor progression and maintenance. And this was validated from another mouse-based paper from the Holland lab. We heard from Frank Szusalski yesterday. They used a Nestin TVA-based system, which showed that a number of different YAP fusions functioned in essentially the same way, that they required binding to TEAD and drove a TEAD-based transcriptional program. And what was interesting with this Nestin TVA-based system, they were able to show that YAP-TP3 can cause brain tumors, which shows that interestingly, this fusion protein can transform cells that are not endothelial cells or endothelial precursors. That has a lot of really interesting implications for tumor biology. We did use these xenograft systems. This is work presented by Keith yesterday, and Nicole Merritt was a postdoc in the lab who was also worked on this project with Keith. And what they showed is that the xenograft systems, similar, just to recapitulate some of the earlier findings, that the fusion proteins affected tumor initiation more than tumor progression, which fits with this overall model that we have of the fusion proteins as being necessary and sufficient initiators of EHE pathogenesis. Frank's work further showed that the C-terminus is important to YAP fusion protein function in the way of chromatin binding. We found similar results, and what Keith presented yesterday was basically work showing that in addition to facilitating chromatin binding, the C-terminus of these fusion proteins are also critical to recruiting epigenetic modifiers and altering the chromatin landscape, which then subsequently modulates the final transcriptome for both TAS-CAMTA1 and the YAP-TP3. And what's great with all of these models is that it's giving us these platforms to dissect in greater detail how these different therapeutic approaches that we're starting to see in patients with these clinical trials that we heard from yesterday. And so this is, I think, going to be a powerful approach. We're going to see how trametinib, which is a MEK inhibitor, eribulin, a microtubule inhibitor, and then these really cutting-edge TEAD inhibitors, what kind of effect they'll have in EHE tumors in actual patients. And then because we have these fantastic mouse models that have been developed, we'll be able to dissect in greater detail how these drugs are working. And I think that we'll get additional insight into EHE and further therapeutic approaches down the line. I think that we still, we heard yesterday about some ex vivo approaches to study EHE. I think that's also going to be something really critical in the future to validate some of the preclinical data that we're driving these mouse models. This is a EHE primate cell culture that I helped isolate, or that I worked on and isolated in Brian Rubin's lab. And so I think that there's a lot of room to grow in this area as well. Just wanted to thank everyone for their time today. These are the folks that did the work. In terms of our most recent work from the lab, this was done by Keith, who is an outstanding graduate student and graduating from the lab this year. Nikki was a postdoc who graduated from 2018, who also worked a lot on this project. Colleen is a really experienced RA who did a great job with a lot of the NGS techniques that Keith presented yesterday. Want to acknowledge our collaborators. Brian Rubin was a fantastic mentor during my postdoc. Also acknowledge our sources of funding, including I just wanted to point out that we have been successful in obtaining federal support for these EHE studies. And that's it. I think I'm I'm going to stop sharing at this point, and looking forward to taking part in the Q&A session later. Thanks.
Our next presenter will be made by video, Dr. Antonescu.
Hi, good morning. This is Cristina Antonescu. I'm a sarcoma pathologist at Memorial Sloan Kettering Cancer Center in New York. And I am very happy to be invited to present at this forum. I was asked for this meeting to cover a topic on molecular testing and how that impacts in the diagnosis and prognosis of epithelioid hemangioendothelioma. So I will go over mainly 3 relatively new genomic methods that we provide here at Sloan Kettering. I will describe them in more detail. These are first the Archer testing, which is a targeted RNA sequencing platform, which is ideal for picking up translocations. The second method is the Msk Impact, which is a platform which is best for picking up mutations and copy number alteration. And the last platform is the Msk Access, which is another sequencing platform that identifies cancer drivers in circulating tumor DNA that is found in the plasma cell-free DNA. So I'm going to start first with the Archer method or platform, which is a targeted RNA sequencing. This is, as I mentioned, an ideal method to pick up gene fusions or translocations that are obviously relevant to epithelioid hemangioendothelioma. The diagram of the actual method is illustrated here on the right-hand side. It's actually a very ingenious method using 2 barcode adapters that link to the target gene here in blue, and is followed by a nested or multiplex PCR, which starts the sequencing of the target gene and then crosses over the breakpoint and sequence the unknown gene partner, and thus we are able to identify the gene fusion that tumor has. This particular platform includes probes and primers for 123 genes, which would cover most frequent translocation described in solid cancers. Obviously, Archer is used in diagnosis, as I mentioned, of EHE, and has mostly replaced the former method, which was FISH, fluorescence in situ hybridization, which would look at one gene partner at a time. So Archer provides information about both gene partners and typically require a relatively small amount of tissue, works in paraffins or archival material, and the turnaround time is about a week. So using this method, we obviously can identify the most common fusion in EHE, the WWTR1 CAMTA1, which comprises about 90% of all fusions detected in EHE, and this correlates with the classic variant EHE under microscope. And then the second fusion is the YAP1-TFE3, which occurs in 5 to 10% of epithelioid hemangioendothelioma, and this correlates with the different pathologic variant under the microscope. By using this method, we were also able to pick up rare molecular variants which I'm going to describe in a second, involving either WW3R1, MAMEL2, or YAP1. So is fusion detection important in EHE? Obviously, it is important to confirm the diagnosis. Sometimes the EHE diagnosis can be challenging, and distinguishing from other vascular tumors or when the material is very scant. So molecular testing is paramount to first confirm the diagnosis. But second, the fusion type is also important for prognosis, and as we have shown in a recent study published in Modern Pathology, as well as even more recent study from Brian Rubin's group, the EHE with the YAP1 TFE3 fusion seems to have a more indolent outcome, despite the fact that some patients may present with multifocal or even metastatic disease. YAP1 TFE3 follows a more indolent outcome, and here you can see this Kaplan-Meier curve in comparison with a more frequent WW3R1 CAMTA1 fusion, the 5-year overall survival being 86% for YAP1 TFE3, while the 5-year for WW3R1 CAMTA being 58%. If we look only at EHE with WW3R1 CAMTA1 fusion, the outcome is driven based on the clinical presentation and the visceral location, with pleural location having the most unfavorable, while the soft tissue and liver having the best outcome. As I mentioned before, these genomic NGS, next generation sequencing platforms such as Archer have allowed us to identify rare cases with novel translocation. And in a recent publication, we described this WW3R1 MAMEL2 fusion that seems to correlate with a cardiac EHE, either in the left atrium or the right ventricle. And here are some microscopic images to show the overlap with a more conventional epithelioid hemangioendothelioma. These particular patients that have this rare alternative variant had an aggressive clinical outcome. Moving on to the second platform, which is Msk Impact, which is a targeted DNA sequencing platform. And this slide here summarizes the workflow. First, the patient needs to be consented on an IRB protocol. So only patients that are being seen at Memorial by a Memorial physician are able to be consented and get this test done. So after the consent, then we would need tissue from the tumor as well as a blood sample. These are accessioned separately in our system, and then DNA is being extracted from both the tumor and the normal blood. The DNA is being pooled, and we use hybridization-based capture, and together with some biotinylated probes, the sample preparation is being done. And the next step will be to have the sample tested through the sequencer. And then once the sequencing is being done, then it runs through certain bioinformatic pipelines. And then the results are being ready to be reviewed by the molecular attending and the bioinformatician. And then once the list of mutations and mutations is being generated, then this is cross-referenced to make sure that only or mainly the mutations that are known pathogenic and oncogenic in cancer are being listed in a clinical report. So this particular test includes about 500 cancer genes that are being sequenced. It also may allow the sequencing for structural variation of a limited number of fusions, but it's not relevant for epithelioid hemangioendothelioma fusion, as well as may provide information about copy number alteration. Okay, moving on to the last platform that we offer here at Memorial is the Msk Access. Here we do sequencing on blood, on a blood sample, not on the tumor tissue. So we take advantage of the circulating tumor DNA, which is found in the plasma. So here this diagram shows that a blood sample is taken from the patient, is centrifuged, and then we separate the white blood cells from the plasma here in yellow, and this plasma would contain a cell-free DNA, which is then being sequenced at a very high coverage, up to 20,000 coverage. And this particular platform includes sequences 129 genes, as well as detects structure variation in 10 genes, mainly for the kinase fusions. Unfortunately, this does not look for the EHE translocations. This is a great method in order to look at different time points to identify if there is any relapse or if there is any progression in the mutational landscape after different clinical trials or chemotherapy. Okay, so let's look to see about the EHE samples at Sloan Kettering. Here is a oncoprint that summarizes the EHE samples that have been tested by Impact. So this targeted DNA sequencing that includes about 500 cancer genes, and you can see here that only a small subset, about, I don't know, 12% of EHE samples show a second site genetic mutation, either P16 deletion or an ATRX mutation and so forth. So, and these are again only what we call the OncoKB mutations that are well known to be pathogenic in cancer. And these don't include other mutations that have been identified, but their function is not known in cancer. So when we look at the survival in EHE patients, maybe because we have very few events, or maybe because we have very few secondary mutations, we don't see any correlation with survival in patients that have second site mutation versus not, as you can see here in these two graphs.
Looks like that is the end of the video. Let's invite in for the Q&A session, Dr. Lamar, Dr. Tanas, and Dr. Antonescu. You can please join me on audio and video. And we've got several questions in from the audience. And just a quick administrative note for the audience, we are seeing all of your questions. We won't have time to get to all of them with this group, but we are saving questions for the Ask the Expert session later on. So if we're not getting to them now, we'll try to get to them later. First question for the group here, and this really could go to anyone, is do we know anything about whether the gene fusions are hereditary in any way? And I guess that could mean two things. If you have a fusion, can you pass it on to your offspring? Or is there some genetic predilection to obtaining these fusions, increasing the risk of getting them? Anyone have some knowledge on that, on those points?
I mean, Cristina would be the expert on that, but I think these are all somatic.
Go ahead, please, go ahead, and then I'll.
I was just going to say they're all somatic events, and so they're not believed to be hereditary in any way. But,
yeah, I agree. We don't have any information that this is hereditary. In fact, since our Impact DNA NGS platform includes blood, I had the slide, but I guess was cut off. If the patient signs a separate consent, we can look specifically at the blood to see if there are any germline pathogenic mutations. And we did that for the EHE, all the EHE patients that were sequenced at Memorial with the Impact platform. And there was only one pathogenic mutation found in the blood, which I think is completely irrelevant to the EHE. So the short answer is no.
Excellent, thank you. And this next question, I guess, could be for anyone in the group as well. It's a question about when we're targeting some of these proteins with drugs, like the TEAD protein. Do we worry about the fact that those proteins have a normal role in the body? And what can you say about how best to target them to avoid maybe toxicity effects or unintended effects?
Yeah, I guess I can start. So, you know, that YAP and TAZ themselves are so prevalent in many cancers that, you know, pharmaceutical companies are really trying hard to target that interaction with the TEAD family of proteins or the TEADs themselves. And I think we don't know how toxic they're going to be or if they're going to be toxic in humans yet. In the mouse models and some of the experiments that have been done, things look promising that some of these drugs are tolerated by the mice. And we've heard yesterday that there are at least, there's at least one drug in clinical trials that's going to involve some EHE patients. And then there are other YAP-TAZ TEAD inhibitors that are in clinical trials as well. And so I think we're optimistic that targeting the TEADs could work, but we don't know yet. And so that's why it's worth exploring other options as well that would be more specific to the fusion proteins rather than globally targeting this pathway. But, you know, I think if we have an effective drug, it's gonna be used, and then we'll try to combat side effects after that.
The next question is specifically for Dr. Antonescu. The question is, if we're seeing a fusion in a primary tumor, but not in a metastasis, like a bone met, do you have a genetic explanation for that? Is it an issue that you could have two different versions of the tumor, two different tumor types, or is it an issue of not having enough material, say, in the bone met to measure the fusion? What are your thoughts on that?
Very, very, very good question. Very important point. And we have seen over the years cases like that. I would call them discrepant results between two different samples or between two different molecular tests of one sample. So I would say it's probably either it's technical, meaning the DNA was insufficient, or maybe the quality of the DNA being a bone biopsy, uncertain if it was decalcified material or not. So we think that the fusion is the initiating and driver event in EHE should be there in the primary in any metastatic site.
Excellent, thank you. The next question's for Dr. Lamar. You'd mentioned in your talk in analyzing the pathways that metformin might be an interesting target as a drug. And the question is, what can you say about any plans, or what would the pathway look like to formally research that as a therapeutic candidate, like some of the other drugs that have been further along in the process?
Yeah, I mean, I'm not an MD, so you know, we're certainly not involved in any sort of clinical trials with that drug. You'd probably have to ask some of the folks that are treating this disease. And I should also say that our work is very preliminary. We've done this in fibroblasts, and so I'm certainly not encouraging patients to run out and start putting themselves on any of these drugs. You should talk to your doctors for sure. I mentioned those two because they have been used in some EHE patients, and so maybe this is the pathway that they're acting on, but we really don't know yet. And I think we need to learn a lot more about them to see what they're doing.
And a follow on that. So, you know, is it the work in your lab as you follow on to that and you produce data, that's the type of information that then MD clinician would look at to consider doing a clinical trial with that type of drug?
Yeah, I mean, that's the hope, or at least provide some evidence for trying some of these things in their patients. But again, I think we need to really show that they're working in EHE models before we move forward with any of that. It's just very preliminary at this point.
Thank you. And a question came in for Dr. Tanas. Seeking your opinion on the importance of genetic testing for understanding, what drugs to use or how to analyze any individual patient.
Yeah, I mean, I think a consistent theme across multiple presentations has been this idea that there's secondary genetic hits that play a role in tumor progression. And so while the fusion proteins are, the initiating oncogenic drivers, there may be secondary hits that might be targetable. And so you could consider exome sequencing to identify mutations or perhaps deletion events by CGH or other methods. I think the other thing that people have alluded to as well is that looking at transcriptomic analysis in either the EHE mouse models or clinical samples would yield, would identify genes that are differentially expressed, and it could also serve as therapeutic targets. And so a combination of these different approaches could probably be utilized to further tailor patient therapy. So maybe you utilize a TEAD inhibitor, but then target another gene that's being overexpressed downstream of the fusion proteins, or one of these other secondary genetic hits that have been identified. I think another potential for combination therapy would be targeting these epigenetic modifiers that are being recruited by the C-terminus of these fusion proteins. And you can imagine, a drug targeting these enzymatic complexes also being used in combination with a TEAD inhibitor, or another gene another small molecule targeting a gene that's differentially expressed downstream of the fusion protein. So I think there's a lot of opportunities for combination therapies that could be tailored to a patient's individual tumor.
And then one final question for Dr. Antonescu related to that topic. We're seeing all of these different areas that could be targeted along the biological pathway, and we might have a number of different drugs, and as we just heard, possible drug combinations that could be used. Do you foresee that genetic testing will become a standard in this for EHE in terms of selecting the right therapy or combination of therapies for patients?
I think we are already doing it, right? We are doing it for diagnosis, as I mentioned, and we are using the most sensitive method for diagnosing the common fusion as well as variant fusions. So that's at the diagnostic level. And then for your specific question, for precision oncology, to choose the right patients for the right clinical trials, we are doing Impact to identify those secondary hits. And hopefully the Msk Access, looking also at the blood, right? What for each patient after each trial, if of markers of resistance or markers of progression. So that we have the tools already. So this is already a reality. It's all about how many drug options are available there to match all these different pathways.
Excellent. Thank you all for your presentation.